So at this point, the test is on the material that is in the module that says lecture exam 2, right? So if you look at that, we'll go to, and I'll just share my screen. I don't have a webcam that works, so that's why you don't see me. And the first time we did this, I tried to do a laptop and it crashed twice. So I'll just go ahead and make sure I can show everything here. So again, this is a little different because it's ours. My canvas is a little different than yours. So if I go to modules and go down, right, when I look at lectures from YouTube for test 2 summer 2020, right, these are the things that would be on the first exam. So as I said at the beginning, membranes, energy and enzymes, photosynthesis, respiration, and then the 2 parts to cell division. Okay, so that's what we're looking at for material for the first exam. You know, I think, you know, one of the answers was, you know, cumulative. Everything builds on everything else, right? And so we're sort of trying to paint a picture and tie everything together. You'll really see that in the last part of the class where we go back and revisit evolution as an example, because, you know, all the stuff you learned on, for example, meiosis, especially, is going to be very important in terms of when we talk about genetics. And if you don't understand meiosis, it's going to be very difficult to understand genetics. And then if you don't understand genetics, it's going to be very difficult to go back and understand evolution. And so we're going to hopefully tie it all together as well. But you know, you've got to understand this stuff now because it serves as the basis for the things later. So that's why I think that, you know, it's not specifically cumulative. It's unlikely you're going to see a test question on what happens in, you know, interphase 1 or something of meiosis on the third exam, but you have to understand what happens to be able to understand genetics. So putting it all together would be important. So in terms of the study guides, we don't have study guides for the lecture exams like we did in terms of the lab one we had. And we'll probably have a lab one. I don't even know if it's been done yet, but we will post that if we have it. So, and again, just, you know, go back and review that information from the second module that I just showed you. And obviously the other thing to look at is not just the stuff that I show you here, right? But the other information that helps explain those things. So, you know, these little links to the different concepts that tend to be a little more difficult for people are shown down here. Obviously, you want to review those too as well. If we didn't think it was important, it wouldn't be here, right? So those are kind of the big sections to really look at. And then again, you're hopefully you're taking notes. Those would be important. And then obviously the book.
There was a question about G0. So G0 and apoptosis are not the same thing. So cell death is apoptosis, and that actually can be a good thing. So for example, when you were in utero, you did not have 5 fingers on each hand, you had a paddle. But those cells were programmed to die, and as a result, you ended up with distinct fingers. G0 is just what we call G1 when the cell stops dividing. So lots of cells, when they are mature, don't continue to go through the cell cycle. That's true especially of cells like nerve cells. They often don't continue to divide. And so we just have a name for that we call G0. So G0 and apoptosis, or cell death, are not the same thing.
Okay, so a couple other just points if you're not reading the chat, because I know it's a lot uh, to keep track of. The other thing that you really want to concentrate on, and they were hand selected by Dr. G and myself, are the mastering biology assignments, right? Those questions have a direct correlation with the information that we're studying. So certainly you want to go back and make sure you understand those as well. And that should help you in terms of preparation.
So, question about entropy. Entropy is often described as a measure of disorder. So, entropy increases as things become more disorganized. Personally, I actually prefer to think of entropy in terms of the quality of the energy, the usefulness of the energy. So, heat, for example, is a very poor form of energy. So, when we convert one form of energy into another, right? So, the first law says we can't make energy and we can't destroy it, but we can convert it. But when that happens, entropy increases. So, the usefulness of the energy goes down as we convert one form to another. So when you flip on the lights, you're converting electrical energy into light energy, but you're also getting heat. So that's why, LEDs and fluorescents are way more efficient than the old-fashioned incandescent bulbs because they produce less entropy. More of the energy is actually being converted to a form that you want. So entropy deals with the quality of the energy or the disorder, and it always increases. So you're not getting a very good deal, if you will, when you convert one form into another. And yes, the mastering biology should be doable again. So your first submission is what counts towards your grades, but there's no reason why you can't go back and do them again as practice. In fact, I would highly recommend it. How is entropy measured? I'm not sure. James, do you know how they figure out delta S when they're doing an experiment?
I have no idea. It's a good question.
Yeah. Not being a chemist, I can only give a supposition as to how that would be measured.
We also had the question on, so where does apoptosis fall? Not exactly sure what is meant by that question. So apoptosis is a normal physiological happening that is just sometimes referred to as programmed cell death. That as Dr. G said, our body has a number of different things that have to die off in order for us to develop normally, especially during development. Same thing happens with the nervous system as it develops, and neurons from our central nervous system find targets on the periphery, and ones that don't, they just die off, and that's apoptosis. It plays a role in certain types of cancer as well, where if you don't have that programmed cell death, then you could have cancer cells, which then could spread and things like that. So I think that's probably sufficient understanding for what you would have to know for the upcoming exam. So what else, people?
Can programmed cell death occur at any point in time in a cell's life? Then?
Yes, absolutely. But it would have to be a cell that was supposed to die. Does that make sense? So just because a cell dies doesn't necessarily mean it's programmed cell death. You could have cell death from trauma or lack of oxygen, like someone has a heart attack, right? And so they lose, the some of the muscles in their heart don't get enough oxygen, so that heart muscle dies. That wouldn't be apoptosis because it wasn't programmed. Does that make sense? Generally, what's left of the cells get destroyed, and the components recycled. And so, you have the lysosomes and things like that that would take care of it, or some of the information, not some of the information, some of the material could also be destroyed by the immune system as well. And that happens in a lot of cases. As an example, for red blood cells, they last somewhere between 90 and 120 days on average, so 3 or 4 months. And when red blood cells become less flexible, they get consumed by special types of white blood cells that break them down and generally recycle a number of those components, especially like the iron, as an example, because it's tough to hang on to iron in the human body. So we generally just destroy and recycle the materials that for other use of other things. So the body's very efficient that way in most cases. So the question in the chat was about protein accumulating and causing the signal for apoptosis to be ignored. So what is that called? And it's just what happens normally, right? So it's not something that you have a particular name for.
Can you please go over oxidative phosphorylation?
That's a pretty big topic. What in particular would you like to know, or should I go over the entire thing?
The proton gradient's role in.
All right. Find the slides for it.
Yeah, you should be able to share your screen, Rick.
Yeah, I just wanted to pop up the PowerPoint before I did that.
Yeah.
All right, is that big enough?
Yeah, it looks good.
Okay, so oxidative phosphorylation is just using the oxidation, initially of glucose, but more proximally of NADH and FADH2 for the production of ATP. So phosphorylation is any reaction that adds a phosphate. The only phosphorylation that is of significance to you all is the phosphorylation of ADP. So if I add a phosphate to ADP, that makes ATP. That is a phosphorylation. Now, as I'm sure you're aware, adding that phosphate to ADP requires energy. So where do we get the energy from? Well, when NADH and FADH2 are oxidized, so hence the name oxidative, those electrons are high energy electrons. And the proteins, that's what these kind of purpley blob thingies are, the proteins of the electron transport system can accept and then pass on those electrons. So they become reduced and then oxidized in turn. But as they pass the electrons from protein to protein to protein, the energy of the electrons decreases. And it's the energy that the electrons are losing, in a sense, that these proteins are using as the energy source to pump protons, hydrogen ions. So the protons are going from an area of low concentration to high. So it takes energy to go from low concentration to high. Where do the proteins get that energy to pump the protons? The answer from the electrons. And the electrons came from NADH, which ultimately came from glucose. So NADH becomes oxidized, it becomes NAD+. Using that energy from those high energy electrons, the proteins create the proton gradient. Well, if it takes energy to go from low concentration to high, it must be the case that it releases energy when protons go from high concentration to low. But they can't go through the phospholipid bilayer by themselves. They're charged, right? So the two things that control whether you can go through a phospholipid bilayer are size and polarity. And although these are very small, they are extremely polar. They're charged. And the only way really for them to get across that membrane is to go through a protein. So initially they went from low concentration to high through these electron transport proteins, but then they go from high concentration to low through ATP synthase. And as those protons go from high concentration to low, that releases energy. The head of this thing literally spins. So there are molecular models that show that ATP synthase spins like a turbine or a water wheel or something like that. And that energy is used to add that phosphate to ADP. So all of that collectively is referred to as oxidative phosphorylation.
Hi, I have a question about membranes. So in the slides, we had pictures of all of the proteins that are like in the phospholipid bilayer. And I wanted to know what the difference is between receptor and recognition proteins because they look pretty much the same. Is there like a difference?
Yeah, so let me see if I can find that. There is a difference. Do you see this slide here now or no? Do you see the membrane slide?
No, it hasn't changed up on mine.
Okay, let me.
No, I'm seeing the respiration slide.
All right, I gotta go back. Hang on, let me stop sharing.
While he's doing that, one of the questions in the chat was, will the video be of tomorrow's test for use to study? The answer will be, assuming it works and processes, I will get it up as soon as I can. It is recording right this second. I can see it on the upper left of my screen, but sometimes it just doesn't work, and we're doing it locally because if you do it through Zoom, sometimes it takes 2 or 3 days, and obviously getting it on Thursday would not help you in terms of studying. So I'll get it up as soon as we can. So the answer will be, in a perfect world, yes, and I'll get it up as soon as it's done.
Yeah, for some reason I'm having a hard time sharing my screen here. All right, how about that? You see it now?
Yeah, that has a different protein.
Okay, so receptor proteins and recognition proteins are very similar in their structure in that they have oligosaccharides typically that point to the exterior of the cell. But receptor proteins receive signals, and recognition proteins recognize other cells. So let's just, for the sake of argument, say I need a new heart, and you say, yeah, sure, take mine. What would happen if they put your heart into me?
Well, your body would reject it, right? Because it's a different organ.
Exactly, right? So
Yeah.
your heart is perfectly fine, but I recognize self. And anything that isn't self, I'm going to reject. Okay? So recognition proteins recognize both self, but also they can recognize other cells that are adjacent. So as cells grow and they become organs, we don't want those organs to be too big or too little, right? So when cells join next to each other, recognition proteins can do that. Receptor proteins are more for like binding to other molecules, hormones, for example, often bind to receptors in membranes, and then that would trigger what was going on inside of the cell. So structurally, they are very similar, but what they're binding to and what they're recognizing are a little bit different. Okay, so receptor proteins are receiving information, recognition proteins are just that, for recognition. And one of the most important things for recognition is self versus non-self. So in the case of, getting a heart transplant, that kind of sucks because you'd have to find somebody who's pretty close to you, otherwise you'd reject it, and that wouldn't work. But on a daily basis, you are bombarded with viruses and bacteria and whatnot, and your body recognizes those as not being you and fights them off.
Okay, thank you.
So there's a question in the chat about the recordings, right? We'll get the recordings up as soon as we can. And any of the recordings we have previously are up in it's the third module down. So the first two modules are the genetics extra homework that you have to turn in not too far away on July 11th, right? So certainly you probably don't want to worry about that until after the exam tomorrow, but you want to get done. It's worth 16 points. That's fairly, in terms of besides our exams, whether it's lecture or lab, that's probably the biggest point thing we have all semester. So make sure you get that done. But and there's also that extra credit iNaturalist assignment, right? That's there. So those are the first two modules. They're just one line each. And then the third one says important course information. And if you go down to the bottom, you can just, of that module, it'll say Zoom 623, Zoom 629 or whatever it was. So you can kind of see those. So I'll post it there when it's ready. And I'll just link to a YouTube account like those do. And if for some reason it doesn't work, I'll post an announcement on that. So I'll post an announcement and send everybody an email because people asked when it's ready and when we post it. All right, other questions? Anything else?
Do we need to know about the leaf cross sections shown in one of the labs?
You should be familiar with the structure of a leaf and how that structure correlates with the photosynthesis reactions that are occurring. Concentration of something, and say, yeah, it's 1% salt, but that doesn't tell you whether it's hypo, hyper, or isotonic. Those are relative terms. So it needs to be in comparison to something else. Now, in almost all cases, what we're comparing it to is the cell, because that's what matters. Sister chromatids and swapping stuff. Yeah, let me pull up the mitosis meiosis PowerPoint.
While he's doing that, again, I think that the actual mitosis meiosis lab does a really nice job on some of the basics. And you might even want to go and do that again, or hopefully, not the first time, but do it again, and looking at, what the terminology is for homologous chromosomes and sister chromatids and things like that. So I think that lab really goes over it. I think especially if you're a visual learner, maybe just doing it again to make sure you understand it would help, because that's an important part of that. Just looking at it this way, we have really only 5 sections on this exam when you look at it, and if there's not looking at the short answer or the multiple answer questions, right? You've got 40 multiple choice. That means there's somewhere between, probably 5 and 10 questions from each little section. So, if there's, 8, 9, 10 questions on meiosis, mitosis, and you don't understand these important little things, then you're going to struggle on that part. And if you, don't understand, 20% of it, then it's going to be difficult to do well in the exam. So make sure you go back and look at that stuff that we're going to talk about right now.
So that's a great point, and let me doubly emphasize meiosis. So perhaps you know this, my specialty is in genetics. I know more about genetics than I know about other things, and guarantee you will not understand genetics well unless you understand meiosis. The good news is, if you understand meiosis, genetics is a piece of cake. So you have a twofold reason to concentrate on learning meiosis. The first is you have a test on it tomorrow, among other things. And the second is the next big chunk of material will rely upon your understanding of it. So I would highly recommend that you understand meiosis. What I tell students when I'm face-to-face with them is, if you can't take out a blank piece of paper and close your book and draw out every single stage of mitosis and meiosis, you don't know it well enough. You need to be able to figure out what's going on with the chromosomes at every single step of each stage, and why that matters. Now, the most important thing, the thing that will help you distinguish mitosis and meiosis, is what I have shown here, and that is the homologous chromosome synapse or pair up in meiosis. That doesn't happen in mitosis, and the consequences of that are profound. So if those homologous chromosomes pair up, then several things will happen. Now, first of all, make sure we understand this. Homologous chromosomes are the chromosomes that look alike. So let me go back to this slide here. So as humans, you have 46 chromosomes. Mom gave you 23 and dad gave you 23. The first 22 pairs are called autosomes. The last pair of chromosomes are called sex chromosomes. Now in this karyotype here, they're showing one X and one Y. So this would have been the chromosomes of a male. If you're female, you would have 2 X's. So you have 2 chromosome number 8s, one from mom, one from dad. Those are called homologous chromosomes. If we look at a pair of homologous chromosomes, those chromosomes can be duplicated chromosomes as shown here. So if we have a chromosome, so there's one on the left, one on the right, the chromosome on the left has 2 sister chromatids. So you tell me, are sister chromatids identical, similar, or totally different? Let's see the chat. Pull up the chat. Indeed, they are identical. They're copies of each other. So now let me ask you, are the homologous chromosomes identical, similar, or totally different? Is your answer identical to this question, Tenchi, or the last one? Are homologous chromosomes identical?
Give you a hint, one comes from mom, one comes from dad.
Yeah, let me phrase it to you a little bit different. You come from one creepy ass family if they're identical. Because what that would tell me is your mother and father were genetically identical. And call me judgmental, but that's a little bit creepy. No, they're not identical. But they do have the same genetic information in terms of what genes they carry. What's different about them is which versions of those genes they carry. So maybe mom gave you the yes, you can roll your tongue gene, and dad gave you the no, you can't roll your tongue gene. Those are just alleles for a given trait. So homologous chromosomes are not identical, but they do have the same genes in the same order. What's different about them are which versions of those genes, and we call those alleles. So if we look at what's happening in meiosis, the most important event that is occurring is that those homologous chromosomes synapse. So these are the ones that one came from mom, one came from dad. Now, because of that, they can actually cross over, okay? So if they're not sitting right next to each other, if they're not paired up, or synapsed is the fancy biological term, then they don't cross over. Now, this particular cartoon came from an author that tried to make things as simple as possible. So one homologous chromosome, they're showing all of the dominant or capital letters, and the other one showing all of the recessive alleles or lowercase. That's highly unlikely that dad would have all the dominant alleles and mom would have all the recessive, but it's easier to keep track of what's going on. So when these homologous chromosomes synapse or pair up, they physically cross over and exchange material. So that creates recombination. That creates a shuffling of the genetic information that is present, that you're getting from, your mother and your father. So you got one chromosome number 6 from mom and one from dad. When you go to have kids, you're only going to pass on one of those. Which one? Well, flip a coin. But even if it's your father's chromosome number 6 that you pass on to your first kid, your partner's going to pass on the other one. It may not be entirely your paternal chromosome number 6. It's going to have some of the maternal chromosome number 6 information on it as well. So that event of crossing over does not occur unless those chromosomes synapse. And that creates genetic recombination, which creates variation. The other important thing that is a consequence of that is because the homologous chromosomes synapsed or paired up in prophase, we haven't done anything to unsynapse them yet. So in metaphase 1 of meiosis, the homologous chromosomes line up in pairs. Notice again, the author is trying to make things as simple as possible. They only show crossing over on the big pair of chromosomes. They should also have shown it on the little pair of chromosomes. Every pair of chromosomes will cross over during prophase 1 of meiosis. Keep in mind that the figures that we're showing here and the figures in your textbook are for a 2n = 4 organism. If this was a human cell, you would have 23 pairs of chromosomes all lined up. So they do that just to keep the figures simple. Now, if those homologous chromosomes synapse, and they did in prophase 1, we get crossing over and recombination, and then they line up in pairs. So if they line up in pairs, what's going to separate in anaphase is not the identical sister chromatids, like what happened in mitosis, but rather the homologous chromosomes. And the result of that is we end up with cells that are haploid. And that's exactly what we want out of meiosis. Meiosis is producing your sex cells, your sperm and egg cells. Sperm if you're male, egg if you're female. And we need those to be haploid. So meiosis achieves that. Now, because we still have not separated the chromatids, that's why we need to go through meiosis 2. And meiosis 2 looks very much like mitosis. The only difference is we start with a cell that's already haploid. So mitosis and meiosis 2 are both equational divisions where the chromatids are separating. Obviously, in the case of meiosis 2, those chromatids are no longer identical because we've had crossing over. In mitosis, they still are identical. So the end result of meiosis are haploid cells that are genetically unique. Right? You don't want all of your sperm cells to be identical. If you did, you'd have a bunch of clones running around the house when you go to have a family. So we need to have these cells be haploid, and it sure is great to have some genetic variation. Okay? The X and the Y chromosomes are considered homologous, but they do pair up. But it's only the very tips of those chromosomes that cross over, and that information on the very tips is really irrelevant. So if you'd like to think of the X and the Y chromosomes as homologous but not crossing over, that's probably a good way of thinking of it. Okay. By the way, another misconception is, do your liver cells and skin cells have sex chromosomes? Yep, you got it. They absolutely do. Okay. So, you know, your liver cells and heart cells and nerve cells have little to do with sex, but they do have your XX if you're female and your XY if you're male chromosomes present as well. Okay. Every somatic cell is genetically identical, at least unless there's a mutation. So yes. So every somatic cell is diploid with all 46 chromosomes, all 22 pair of autosomes, and the pair of sex chromosomes. So how much genetic information you have matters. And we'll get to that in the genetic section in things like trisomy 21 or Down syndrome. So if you're a male, you only have one X chromosome, and females have two. And there are lots of genes that have nothing to do with sex that are on the X chromosome. And females have two copies of that, and males have only one. So to mitigate against the uneven amount of genetic information, females take one of their two X chromosomes and wad it up into a ball that becomes inactive. And that's referred to as a bar body. Now, it's random as to which one wads up into a ball and becomes inactive, but once the cell has decided which one is going to be balled up and inactive, every cell that comes from that cell will still have that same X chromosome inactivated. So females end up being somewhat of a mosaic, because that happens during the developmental process, where half of their cells have one X chromosome inactivated and the other half have the other. But it tends to be patches, because once that called lionization after Mary Lion occurs, it is passed on to the next cell. There's actually an RNA molecule that ensures that that same X chromosome remains inactivated. And genetics is not on the test tomorrow, but meiosis is. And no, X chromosome inactivation does not affect meiosis. Going back to the questions, actually meiosis will not occur properly unless every pair of homologs crosses over. So crossing over is pretty much guaranteed for every pair of chromosomes. And yes, the female's two X's will also cross over. I think I got all of the chat questions.
All right, so while you're thinking of other questions, a couple of other thoughts. We have mentioned genetics a couple times and that genetics homework. And again, obviously your main focus today in the next 22 odd hours is to prepare for the exam tomorrow. On Thursday, we do have a Zoom session scheduled to look at the genetics stuff, and Dr. G will be with us. But the point being that it's at 9:30, and the reason why is Dr. G is actually officially on vacation, but he's taking time out of his vacation, which, Blue said that he sounded far away. It's because he is. And so it works best in his vacation schedule to show up at 9:30 on Thursday. And like he said, his PhD literally, I think, says genetics in it. So there's very few people around that know more about that than he does. So he'll be with us on Thursday. So we're starting at 9:30 on Thursday, not 10:30 like normal. So just make sure you make a note of that as well. For the macromolecules that can immediately pass through a membrane through simple diffusion, remember, you have to be small and lipid soluble, or mostly non-polar. And so if you look at the macromolecules, right, we have four basic categories: carbohydrates, lipids, proteins, and the nucleic acids. Carbohydrates, proteins, and nucleic acids are all polar molecules, so they cannot pass directly through the cell membrane, but lipids generally can. Depends a little bit on size, and most of the size lipids that will be macromolecules that we talk about would be able to pass through the cell membrane. So a great example would be like a hormone. Okay, so a hormone is a chemical signal in the body where there are two basic types. One is derived from cholesterol, called a steroid hormone. Since cholesterol is a lipid, they are lipid soluble, and they can pass through the membrane and go directly into the cell. And they typically affect production of a protein in some way, because they can go through the membrane directly. But a protein hormone has to have one of those receptors on the outside of cell, because it's made of proteins. And since it is a polar molecule, it cannot get into the cell, so it affects on the outside of the cell typically. So the main answer is primarily it's the lipids that can move directly through the cell membrane. And again, that has to do with the functional groups, again, showing how everything builds on it. And the lipids have functional groups that are made of methyl groups, right? And so that functional group is a non-polar one, and that's why they can go through the membrane directly. All right, so we also had a question about the PowerPoints, and I'll make sure that you can figure out the links to them and everything, and I'll post an announcement from that. So I'll make sure that we can find them fairly easily. So They are there though. They're in each module where you have to go into the module and then post to it. So as an example, let me share my screen. So if you look, this is the photosynthesis in plant cells. This is not in the first set. It's, that was Dr. G's lecture that we went over, right? But if you go to this one, this is in the second set of the module where it says information for lecture exam 2. If you go to the bottom, right, it says resources about these topics. Watch the photosynthesis lecture on YouTube. That was posted in the previous module. And then review the PowerPoint slides. So if you just click on that, it'll open up, and then you'll be able to see the PowerPoint slides that he used for the module. Does that make sense? So they should be there, and if there's not something there, we're not hiding them from you. The idea was just to go into each module and kind of look at that try to make sure you're looking at the stuff you're supposed to be looking at that's going to help you understand better, I think is part of the reason why we did it. So, and if you can't find something, send me an email, we'll figure it out. But we're not trying to hide it from you.
Let me share for a second here. So I'm in the modules. This is test 2 lecture module. Now it's different from the test 2 lectures that are on YouTube. So each of these pages is a page that deals with topics that are on test number 2. And he just showed you the one on photosynthesis in plant cells. So at the end of that is a link to the PowerPoint. At the end of the meiosis and mitosis, there is a link to the PowerPoint. And if it isn't there, then it's in the previous one that talked about a similar topic where both of them are covered in the same one. So that's where you find the PowerPoint presentations if you don't want to look through the lectures on YouTube. So lectures on YouTube, you see the PowerPoints and you get the lecture. Lecture modules, a link to the PowerPoints is included. Receptor recognition almost always have the oligosaccharides on the outside, and they're not going to flip around because that wouldn't be energetically favorable, because the meat of that phospholipid bilayer sandwich is hydrophobic. Anaphase 2, it's just the chromatid separating. It's just like mitosis anaphase where the chromatids separate. But in anaphase 2 of meiosis, those chromatids are likely not going to be identical anymore because of crossing over that occurred in prophase 1.
Or anything else?
There was a question about the figures on page 111 in the lab manual, and this is a common point of confusion. People confuse chromosomes that are duplicated with haploid and diploid, and they have nothing whatsoever to do with each other. So if you have a cell that is diploid, what that means is that you have both homologs present for every chromosome that there is. It doesn't mean that those chromosomes are duplicated or unduplicated. So how many chromatids are present? Oh, thank you, Tim. So this cell here has one big chromosome and one little chromosome. Therefore, it is haploid. It does not matter that these chromosomes are duplicated. This cell has one big chromosome and one little chromosome. Therefore, it is haploid. And it does not matter that there's only one chromatid. The bottom left one has two big chromosomes and two little ones. That's why it's diploid. And the one on the bottom right has two big chromosomes and two little chromosomes, and that's why it is diploid. It does not matter if chromosomes are duplicated or unduplicated. That is an entirely different question than haploid versus diploid. Yes, prophase 1 is when crossing over happens. And yes, so I can't always cover everything, and it's especially difficult in the online format. So if I did not talk about it and it's still in the PowerPoint, then I would not worry too much about that. Concentrate on the things that were covered and quickly glance through those things that were not.
Yeah, your bulk of your time studying should be spent on the information that was discussed in the videos, the information that is found in the mastering assignments, right? Because that covers most of the stuff that was talked about in the videos. Not everything, but relates to that and your understanding of it. And then that second section that Dr. G just showed you with the links to the PowerPoints and things like that. So that's kind of what you should be paying attention to and focusing on. I wouldn't completely ignore a slide, because usually the slides are there to give some sort of framework to some of the other information, right? But as we go through it to make the video, sometimes obviously, one slide is more important than the other, and trying to keep the information down from making them, a 2-hour video every time, then, we have narrowed down the stuff. So the most important stuff is the stuff that we've talked about and asked about in mastering, and put links to other things on to help you understand that. Anything else? Anybody? All right, so remember, we'll be around for email, mostly me, because Dr. G shouldn't look at it too much because he's on vacation. But we should be able to answer between us anything. So as always, if you do have something to say or a question, then email us both through the regular email, because it just makes it so much easier than going through Canvas to respond, and we both get it, and we both know we got it. Otherwise, make sure you're logged in before 10:30 tomorrow to do that. And then, as Dr. G just posted in the chat, he'll see you on Thursday at 9:30 to discuss genetics. All right, so obviously when you're done with the exam tomorrow, take a look at that genetics if you haven't yet, and start working on that, so you have some questions or at least some knowledge, so when he talks about it, it'll make sense on Thursday. So after the exam, start working on genetics. Anything else? All right, thanks all for the compliments. And then no more questions. I'm gonna end.
Good luck, everybody.
All right, good luck tomorrow. Let me know if you have questions. See ya.
Thank you. Goodbye.
Bye.